So once we have these cells in our body, right, so we have created a liver cell. Let's say we have taken our stem cell has differentiated, and it's turned into a liver cell, or it's turned into a skin cell, or a stomach cell. Then what we have is a whole lot of dividing. And we do that through mitosis. So mitosis is the way our cells divide to make more cells. Now, the cells that go through mitosis are called somatic cells. Somatic, soma, is related to body. So these are all of the body cells. The cells that are not included in this are what are called the germ cells, or the gametes, or the sex cells. So gametes like sperm and egg. So the cells that are going through mitosis are our somatic cells. All of the rest of the body, every other cell in the body except for the sex cells, or the gametes. And the reason that this does this is it's necessary for growth. So if we think of a child being born, they're very, very small compared to an adult. And so those children's bodies are going through mitosis to get bigger, to grow. We need it for healing. So as we're going through life, even if it's something like a cut, or if it's something that we're putting in our bodies that's damaging internal cells, we go through mitosis in order to heal our body. Also to replace cells. So our cells get worn out, they get old, they don't function as well as they used to, so our body will kill them, recycle the parts, and then through mitosis we get new ones. So another important thing to remember about mitosis, not only is it that they are only in somatic cells, it's also that when they divide, they produce cells that are identical. So we start out with a cell with its nucleus, and we've got all of our DNA in here in the nucleus, and then at the end of mitosis, we end up with two what are called daughter cells. And these two daughter cells are identical to each other. So again, this is after we've already differentiated from our stem cell, and we're saying now we have a skin cell. This over here is our skin cell, and then as we damage our skin, or as our skin is growing bigger, then we are making identical skin cells. So some cells are going to divide quite often. So for example, our skin cells, again, they divide quite often. We're sloughing off skin cells as we walk, as we move around, as we touch things, and so we are continuously producing new cells. So some divide pretty much continuously. So some are growing or dividing continuously. Some cells stop dividing after they mature. So for example here, let's see, this would be our skin. For example here, this would be like our neurons in our brain. Again, we don't have a lot of division. Once that cell is matured, it doesn't go through dividing anymore. And then some stop and then start again. So they stop dividing temporarily. But will divide again if necessary. Or those that stop dividing temporarily would be something like a liver cell that hangs around for a while, or bone cells that hang around for a while, but they may need to start dividing again if there's a break in the bone, for example. So that's kind of the general overview of mitosis. Some other important things to note about mitosis is that when we go through mitosis, it's actually just a piece of an overall cell cycle. So I'm going to draw a kind of pie chart here. And what we'll see is that about 10% of the time, this is about 10% of the time, over here this is our 90% of the time, about 10% of the time is when our cells are going through mitosis, which is cellular division. And a part of mitosis here, it's actually kind of at the end here, right after mitosis, is called cytokinesis. And we'll talk about those in just a moment. The bulk of the time over here, this 90%, this is called interphase. And this entire thing is called the cell cycle. So here, this part here in interphase, this is where the cell spends most of its time. This is where the cell is kind of hanging out, being a skin cell, for example, just metabolizing. So it's hanging out, it's doing skin cell stuff, making the proteins that skin cells make. And then also in interphase, it's replicating its DNA. And what that means is it's making copies, or making a copy. I should be more clear. It's making a copy of the DNA. So it's taking the DNA that it has in the nucleus, and then it's making an exact copy, an identical copy of the DNA during interphase. And that's going to be kind of this part of interphase over here, is where we have our DNA replication. And then a little bit more, it's going to get bigger. It's going to replicate other organelles that are important for cell division. And then it will move into mitosis, but it spends most of its time in interphase. So we have our cell cycle, we're spending our time in interphase, but then we get to a point where we need to divide, and then that's when we enter mitosis. Again, in mitosis, we're going to start out with our one cell, and inside that cell is where we have our DNA. And I'm drawing the DNA the way I am because that's generally how our DNA hangs out for the most part during interphase. During interphase, the first part of interphase, our DNA is kind of like if you took a whole bunch of yarn and unraveled it and then threw it in a pile. And the reason it is like that is because we need to get into these parts of the DNA in order to access certain parts of the DNA to make proteins. And we'll talk about that in a moment as well. So our DNA is just kind of hanging out. Through the process of mitosis, though, in first interphase, we're going to make an exact copy of all of our DNA. So we make an exact copy of the DNA. When we do this, so we make an exact copy of the DNA, then we go through the first part of mitosis. The first part of mitosis is called prophase. And during prophase, we're going to see some condensing of our chromosomes. So it may be something like this if you've seen chromosomes before. We'll have little ones. Now, what I mean by chromosomes is that our DNA is split up into pieces. It's not all one big long piece. We actually have 46 different chromosomes. And what I mean by different is individual pieces. They are very, very similar. Some of them are very similar, and we call those that are very similar our pairs. So we have 46 total chromosomes, which ends up being 23 pairs of chromosomes. And what that means is that we actually got 23 chromosomes from our mother, and we got 23 similar chromosomes from our father. So we got 23 chromosomes from mom, 23 chromosomes from dad. Now the chromosomes that we got from mom, those 23, are very similar to the chromosomes we got from dad. So they carry similar information. On one chromosome, let's say, that we got from dad, this codes for eye color. Now, dad's eye color in this part of the chromosome, for example, what this is this is our DNA all wrapped up and condensed in a really small package. And so this chromosome, this condensed DNA package, this area, we're pretending, is coding for our eye color. Now, we have that same area on the chromosome that we got from mom. That area codes for eye color. Now, perhaps, though, they're different in the way that they are coding. Let's say mom is going to code for blue eye color, and let's say dad codes for brown eye color. And so then, when we have these two different genes, as they're called, genes, the gene is what is coding for eye color. So the actual pieces of DNA, like if we were to take our DNA and unravel it and stretch it out, just like we talked about with our nucleotides. Right, we have our A's and T's and G's and C's on one side and A's and T's and G's and C's on the other side. We would say that from this nucleotide to this nucleotide here, this is called a gene. This is the actual information that is going to code for something. And the example here that we're using is eye color. So we have our gene, say this nucleotide to this nucleotide, which in reality are hundreds of thousands long or thousands long. So this gene right here is coding for eye color. We got one gene for eye color from mom. We got one gene for eye color from dad. And this goes on with all of our different genes, everything that codes for every single protein. So our genes actually code for proteins. We have the nuclear information in our DNA. And then that information is stored in our DNA in chromosomes. We went into some gene expression here is what we're talking about. Then what happens is that as this cell is living and being in interphase, it's going about its daily thing, right? It's metabolizing, it's taking in energy, it's using energy, and what it's doing is it is going through gene expression. We'll get back to mitosis as well. But gene expression, meaning that it expresses those genes. And how it does that is by taking the DNA, and we've already talked about this, but turning it into or making a sort of copy of it in RNA. And then we're going to take that RNA, so we're taking this gene here that we said, these nucleotides here code for eye color. We're going to make a sort of copy of it, which is a single-stranded RNA strand. When we make that single-stranded RNA strand, that's also coding for that information. We've just used the DNA. We've opened up the DNA. We've gone in and we've said, okay, we need to make the protein for eye color. So when we do that, we make our copy. Our copy molecule is this RNA strand. And then this RNA strand is going to go out into the cytosol. That is not how you spell cytosol. Cytosol. When it goes out into the cytosol, it's going to meet up with a ribosome. When it meets up with a ribosome, then it's going to make our protein sequence, right, our amino acids. And then it'll go through the steps of folding up to be our protein. And that protein then is, let's say, our brown eye color protein. So what I just outlined here, kind of in this bottom part, I went off on a tangent of gene expression here. We were going to get there anyway, but our gene expression here is taking a gene, which in this example is eye color, but could be anything else, skin color, hair color. I know I'm using all different colors, but how to code different things for the body. And then what we're doing is we're taking that DNA, using it as a template, making RNA as our template piece, bringing that out into the cytosol, meeting up with the ribosome, and turning that into our protein. And that is how we express a gene. And that's what our DNA is coding for. Our DNA is coding for all of the proteins in the body. So it's important that we go through mitosis so that all of our skin cells are coding for the same thing. We've gone through our stem cell step of the process where we have specialized. We now said, okay, we are a skin cell. And now that we are a skin cell, we're going to go about doing skin cell things, which means we are going to go through gene expression, and we're going to be making the kinds of proteins that we make in a skin cell. And as we're doing this, right, in interphase, we're being a skin cell, making skin cell proteins, then we're going to go into our DNA replication part of interphase. Once we do our DNA replication part, then we're going to move into mitosis, and that's where we're going to meet back up here at the top and talk some more about mitosis. So mitosis, we're taking our 46 total chromosomes, right, 23 from mom and 23 from dad, and they are matching chromosomes. So again, we have the same types of genes on those chromosomes. We get one set from mom, one set from dad. So we have 23 pairs and 46 total. These are called homologous. Homologous chromosomes. And we'll revisit that when we talk about meiosis. But our homologous chromosomes, because they have similar information. Not exact, because it could be blue versus brown eye color, but nonetheless, they both code for eye color. So as we are moving through mitosis, we're going to take the DNA, as I mentioned, and we are going to condense it. And so we take it from this kind of yarn that we've just thrown in a pile, and we condense it into these nice little, what we call chromosomes, because it's all packaged up and condensed. So then what we do is we are going to move from our stage where we have condensed our DNA, and mitosis again is when we're dividing up our nuclear information, dividing up the DNA. So in mitosis, we are looking at somehow packaging up and moving around the DNA. And we do that by condensing the chromosomes there. And then once we condense the chromosomes, we're going to divide them up. So we actually line them up at the center of the cell. This right here, this is one sister chromatid. Technically, it's called a sister chromatid, and this over here is another sister chromatid. Now, it used to be called a chromosome. So way back at the beginning here, when we talked about that one cell that we got from mom, we took that and we made a copy of it. Remember, we made an exact copy of it. When we did that, the copy sticks to the other one. So that's how we get this butterfly-looking thing. And then we had that same chromosome, or similar chromosome, that we got from Dad, and then that also replicates. We make a copy of it, and it stays stuck to it. So then what we have here are our chromosomes that have been duplicated. So the same thing is on the left as is on the right in these little butterfly guys. Okay, so we've lined them up in the middle of the cell here, and then we're going to pull them to opposite sides of the cell. We're going to divide the information. We're going to do that by ripping these things in half, because what we're doing is we're ripping apart the copies. So we will see that over here. So we move into the next stage. We've pulled apart our copies. So now what we have, I'm going to stick with the colors now, is we have one of the big red ones that we got from over here on the left side, one of the big red ones on the right, and then we have a small red one on the left, and we have a small red one on the right. We have a big blue one on the left, a big blue one on the right, a small blue one on the left, and a small blue one on the right. So you see how we have divided up our chromosomes. We've ripped one copy to the left side, and we brought one copy to the right side. So now we have separated our chromosomes. Now that we've done that, then the cell is going to go back into being a nucleus. So what we see is that we have this in duplicate now. So over here we have our cell envelope, or nuclear envelope rather. Nuclear envelope is going to start to form around these chromosomes. Let me draw our chromosomes really quick. So they're going to get all nice and close here. Still kind of far apart, but. And then our nuclear envelope is going to form on opposite sides of the cell. Now I've simplified this because, right, we don't have just 2 red chromosomes and 2 blue chromosomes. We have 23 red and then 23 blue for our total of 46, right? But for the purposes of the drawing, I've simplified it, 2 mom chromosomes, 2 dad chromosomes. And then we have the exact copies that we've now pulled to opposite sides of the cell. And then once we have built this nuclear envelope around those chromosomes now that are separated, we have an exact copy or an exact set over here compared to this set over here. They're exactly the same. Then we are done with mitosis and we move into something called cytokinesis. Cytokinesis, cyto meaning cell, and kinesis meaning movement and meaning splitting here. So we are going to be breaking the cell apart now, the cytosol. So what happens is we see that there's what's called an invagination here, where this is going to start to come down. This is our plasma membrane, is going to start to come down, and it's going to move more and more to the center of the cell until what happens is they are pinched off, and now they are two separate cells. So we go down the center, and then we end up with one cell here, with our nucleus and our nucleus. And then, of course, we have our chromosomes, our big red one, small red one, big blue one, small blue one. And now we have two identical daughter cells. These are daughter cells. And this is mitosis. So again, remember that mitosis is specific to our somatic cells, all of our skin cells and hair cells and liver cells and all of these types of cells. And then we have what's called meiosis that we're going to talk about briefly as well. And in meiosis, what we're talking about are those cells that we had as the exception earlier. So we're talking about the gametes. Okay, so let's take a closer look at meiosis then. So meiosis. So meiosis is used for sexual reproduction. Okay, so these are our gametes. And when we say gametes, what we mean is the specific sperm cell and the ova or the egg cell in the females. So this is a special process that's not exactly like mitosis, because if we think about what mitosis does just for a minute, remember mitosis took our 46 chromosomes, our 23 pairs that we have in each cell, and it made an exact copy. Now, if we did that in a sperm cell, for example, or in an ova, an egg cell, for example, then what we would be doing is the father would be passing on 46 chromosomes, and the mother would be passing on 46 chromosomes. Now, when we put that together, we've got way too many chromosomes, right? So we're looking at 92 chromosomes in that new child that's going to be born, the zygote. Then, if you can imagine the next set, let's say they now have 92 chromosomes, which isn't possible, but let's say they do, and let's say they meet up with someone, let's say this is the dad here, so our sperm cell, and then they meet up with someone, mom over here with her ova, and she has 92 chromosomes because her parents each had 46 and went through mitosis and passed those on. Then we're getting up into bigger and bigger and bigger numbers, and so this does not work. We need to have a way to maintain our 46 chromosomes, our 23 pairs. And we do that by going through meiosis. In meiosis, we still have interphase. Okay, in our cell cycle, there's interphase where the cells are hanging out, metabolizing different things. They do go through the process of DNA replication. So actually, let me do this first. So we have interphase. And in interphase, they're metabolizing, our fancy word for just going about doing cell stuff day to day. And then they also replicate DNA. So we are going to make a copy of the DNA in these cells as well. So we're going to just talk generally about meiosis and not get into too much detail with meiosis. But we still have the same situation. The DNA is kind of all hanging out. Then we go through the process. At this point already here, this is after interphase, so interphase is over here. Our DNA is still hanging out, but it's duplicated. Then we're going to condense it. So condense our duplicated DNA. Remember, we have our chromosomes with their sister chromatids. And we have our copies there. Now, one of the differences that we see in meiosis compared to mitosis is that rather than all of these chromosomes, so our nuclear envelope is going to break down, it's going to release all these chromosomes into the cell, and instead of them lining up like we saw in mitosis, they are going to line up with their homologous pair. So they're going to hang out right next to the copied one that has similar information. So remember that in mitosis, these were all individually lined up along the cell. And what we see here is that they're lining up with their pair. Then what happens is we pull the pairs apart. like so. So what we are bringing over here is the duplicated red ones here and our duplicated blue ones here. And then it actually is going to go through the process of dividing up the cell similar to what we saw in mitosis. So we have the nuclear envelope on either side. We're going to go through. We have the invagination here. And it's going to split these up into the two cells. Now at this point we have a duplicated chromosome over here and duplicated chromosome over here. All of these are duplicated still. So then what happens is in meiosis we actually go through another round of division. So we have 2 rounds of division. So now we're taking these cells, and what we're going to do is we are going to take each cell, and they are going to go through their own division here, our second round of division. So we're going to see our chromosomes now line up on our metaphase plate or in the middle of the cell. And just like we saw in mitosis, we're going to then see a division or a splitting up here. We're going to pull the chromosomes to opposite sides of the cell. And then we're going to see the same thing that we saw in mitosis and just now in what's called meiosis 1. So it's actually called meiosis 1 and meiosis 2. Meiosis 1 is when we had that first division that ended us up right here. If I can make that clear, this is the end of our meiosis 1. And then we moved into here, meiosis 2, which is our next division. So we have our chromosomes, our duplicated chromosomes lining up in the middle of the cell, and then we pull them to opposite sides of the cell. Then what we do is we create a nucleus, a nuclear envelope around these things. Okay, and then we see that same type of invagination here. Our cell membrane is going to start pulling into the center of the cell until it pinches it off into 2 cells. Same thing here. Go down here, and what we get are 2 cells from this as well. So we have our 2 cells with their nuclei, and at the end what we actually have is 4 cells, and it has half of the information. Try to draw my chromosomes here. There we go. So now we have 4 cells. So 1, 2, 3, 4 cells. And we started, remember, with 1 single cell. Okay, we started with our single cell right up here. Well, I guess I didn't scroll over enough. Our individual single cell was all the way up here when we started, and then we went through the process here. And remember that our original cell, our original cell started with our 46 chromosomes that had been duplicated, right? So they're duplicated here. That's why we see the butterfly kind of thing. And then we go through the process of separating those duplicated homologous pairs, those chromosomes are homologous pairs. We separate those. That's all in meiosis 1. Then we moved into meiosis 2. And we took those cells that we had just divided, so we had 2 cells, and both of those cells are going through another division, and then at the end we end up with 4 cells. And so we see this happen with sperm cells. We have 1 starting sperm cell that is going to divide 2 times, meiosis 1, meiosis 2, and end up with 4 sperm cells that have half of the chromosomes. In this example here, if we go back to the first cell, in this example, of course, remember we have 46, but in this example right here, we had 1, 2, 3, 4 chromosomes, right? 4 total chromosomes, 2 pairs, right? This was a pair here, this was a pair here, 4 total chromosomes. When we get to the end here, and we look at our 4 cells that we get in the end, notice that there are 2 total chromosomes, half of the chromosomes, okay? And there aren't any pairs. They're all individual chromosomes. We're not sending any pairs to anybody. They're individual chromosomes. And all of these cells, we won't get into the specifics, but all 4 of these cells are different. Okay, and that's from a couple of different things. We're not going to get into the details in this class. It's just important that you know that we have an original cell in real life. In humans, it starts out with 46 chromosomes, 23 pairs. We go through 2 divisions to separate the DNA. And when we do that, we end up with 4 cells at the end that have half the number of chromosomes. So instead of having our 46 chromosomes, now they have 23 total individual chromosomes. And this is, again, the sperm cell and the ova, which is the egg cell. Okay? You should just know these kind of basics about mitosis and basics about meiosis. Our mitosis are somatic cells, 1 division, 2 identical cells. Meiosis is our gametes, our gametes, sperm cells, ova. They go through 2 divisions. At the end, we end up with 4 cells, and they are all different cells. That's another important piece, is that they are all different cells.